Hello everyone, welcome back to the channel Bio-Microbio-Logy. I'm here again with a new topic, bacterial growth curve. Throughout the entire video, we are going to discuss about the pattern of microbial growth. In natural environment, bacteria or microbes obtain their nutrients by their own and increase their population. When we want to do the same in the laboratory, they need to provide everything like proper pH, temperature, nutrients, oxygen or other requirements for their growth. So if we want to know their growth pattern, we need to culture the bacterial sample in a proper manner in the lab, and at the end of the culture we have to analyze their growth pattern. So what is growth? Growth is the natural phenomenon, also a fundamental characteristic of every living system. In each living system, growth happens gradually. Sometimes growth can be a development, sometimes can be an improvement, sometimes it can be an increase in the population. Like every living matter, bacteria multiply in a manner of increase in cell number by cell division is called binary fission. A figure of cell division at its simplest form is drawn in below. Here I took an example of understanding growth. In multicellular organism, an increase in the size of individuals indicates its growth. Like when a baby of human grows, it becomes an adult by increase in the size and development, not in number, though there is a continuous increase in the cell number as it's a multicellular organism. On the other hand, in case of unicellular organism, an increase in the cell number indicates their growth. Likewise, bacterial cell grows by asexual reproduction of cell division, which results in the doubling of cell number. Now the question is how bacterial cell multiply. Bacteria multiply by a process called binary fission. That's a process where cell elongates, replicates its own DNA, cell divides and pass a copy of its DNA to the divided half of the cell. Binary fission takes place at a specific time interval called generation time or doubling time. Suppose a bacteria starts dividing from a single cell. After its first generation, it becomes double in number. And bacteria grows in a geometric progressions like 2 to the power 0, 2 to the power 1, 2 to the power 2, 3, 4 up to the power 2 to the power n, where n is the generation number and each generation it doubles its number. That's why it refers to exponential progression of growth. The figure is showing the geometric progression of bacterial cell division by binary fission. Now we are coming to the bacterial growth in batch culture. Bacterial growth is studied by analyzing the growth pattern from the growth curve of bacteria. To know the same, bacterial sample is cultured in laboratory. The sample is grown in a closed vessel. Either it can be a conical flask or stirred tank under a variety of physical, chemical and nutritional conditions. In the way, the bacteria is cultured in a single batch medium in the closed vessel at a specific time interval for the incubation, where no fresh medium is provided during the incubation, is called batch culture. So, what is bacterial growth curve? As because the bacteria is incubated in a batch culture in a single batch medium where no fresh medium is provided to the culture during incubation, the growth occurs in its own manner, and from the culture we get a typical growth curve of bacteria that suggests a pattern or phases of microbial growth. Bacterial growth curve can be plotted as the logarithm of the number of bacterial cell versus incubation time, having 4 distinct phases: lag phase, log phase or exponential phase, stationary phase and death phase. The figure is showing a typical growth curve of bacteria. At the initial phase, there is no progression in the growth curve. In this phase, it's assumed that bacterial cell might be evolving the new environment or the inoculum size is less sufficient for dividing or growing, or maybe the sample of bacterial cell is activating its enzyme for growth and metabolism. That's why this phase is called lag phase. In the next phase, it's showing a sudden and rapid increase in growth curve. An uplifted line is showing here, where the bacterial cell divides rapidly in an exponential manner and utilize the nutrients at hide it, and the population size reaches at maximum level. That's why it's called log phase or exponential growth phase. Just after the log phase, the growth curve becomes horizontal. It indicates that the overall cell population reaches at a point where no more nutrients available, as well as the environment becomes unfavorable to grow for the other cell which are about to divide. I am saying that because every cell of bacteria have different time for division or different generation time. As a result, some cells die which completes generation number, and new cell divides newly and growth rate becomes equal to the death rate. That's why it's called stationary growth phase, where no that much of growth is observed. And after that, some cells are usually dying and the population size is decreasing because of the cell death is increasing. Now we are coming to the mathematical expression of growth curve. If we want to express mathematically the growth pattern of bacteria, then we have to take the initial number of cell is n0, and after t time and n generation number, the cell number will be nt, and the relationship is nt equals to n0 into 2 to the power n, where t is the generation time. By logarithm of both sides of the equation nt equals to n0 into 2 to the power n, we get log nt equals to log n0 plus n log 2, or n log 2 equals to log nt minus log n0, or n equals to log nt minus log n0 divided by log 2. Here value of the log 2 is 0.301, or n equals to log nt minus log n0 divided by 0.301. This is the mathematical expression of growth curve.